So, it bacteria is a nga prokaryotic cell. It bacteria na acquire food tikang ha organic matter. Then, may dalawang mga bacteria nga consider nga parasitic, meaning kailangan ni rin usa nga host organism para makapagprodusirahin pagkaon. Then, asya adtoiraan mga nakos hindi diseases. May daliwat mga bacteria nga nakakapag-provide ran ira own food or nakakapag-produsira through photosynthesis. Bacteria are divided into categories according to their physical characteristic. So those bacteria that are gram-positive and have a cell wall, gram-negative and have a cell wall, and those that lack a cell wall. So characteristics of bacteria are examined to provide data for identification and classification. So those characteristics include the cell morphology, the staining reactions, motility, colony morphology, atmospheric requirements, nutritional requirements, biochemical and metabolic activities, specific enzymes that the organisms produces, pathogenicity, and the genetic composition. Cell morphology is ginagamit para ma-determine ang size and shape nganhan morphologic arrangement han usa na bacteria. So, ha size, it bacteria is na-range yahin 0.2 micrometer in diameter up to 10.0 micrometer long spiral shaped bacteria. So, may the shapes hit bacteria. Kung it is round or spherical bacteria, tinatawag yan na cocci. Kung rectangular or rod-shaped bacteria, bacilli. Kung curved ng spiral-shaped bacteria is the Spiralia. So, it cocci, pwede single or in pairs. Tinatawag ito yang diplococci. Kung chains of cocci, streptococci tawag. Kung clusters of cocci is staphylococci. Kung packets of 4 cocci, it is called as tetrads. Kung packets of 8 cocci, octads. So, may mga methods or staining techniques na ginagamit para ma-observe ang usa nga bacteria cell morphology. Like, for example, ang iya size, ang iya shape, ang iya morphologic arrangement, ngan in iya composition ang iya cell wall. Usa nga technique na ginagamit is an gram stain, in which it differentiates an gram-positive ngan gram-negative han cell wall han usa nga bacteria. So, kung gram-positive iya, at the end of the procedure, it iya color is either blue or purple. So, it gram-positive is a thick layer of peptidoglycan ha cell wall. Then, kung gram-negative iya, it iya color at the end of the procedure is it's either pink or red. It gram-negative is a thin layer of peptidoglycan. So, madali lang iya ma-remove. Unlike gram-positive. So, next is the motility. So, it is the ability usa nga bacteria to swim. So, ang bacteria nga may dahini nga ability is tinatawag nga motile. Pero ang mga bacteria who are unable to swim are said to be non-motile. So, ang reason kung kaya nakakaswim sa nga bacteria is through the presence ng flagella or an iya axial filaments. Next is the colony morphology. So, bacterial colony Those are piles of bacteria. So, it colony na contains siyahin millions of organisms. So, it appearance it usa nga bacteria is na vary from one species to another. So, it colony morphology is an important clue para ma-identify an usa na bacteria. This is useful para ma-classify an basis of relationship han usa nga bacteria to oxygen and carbon dioxide. So, bacteria is made the 5 major groups: obligate aerobes, microaerophilic aerobes, facultative anaerobes, aerotolerant anaerobes, and obligate anaerobes. So simply, it obligate aerobes is kailangan niyahin oxygen. It microaerophilic aerobes is kailangan gyap nirahin oxygen pero in a lower concentration. So they prefer an atmosphere that contains about 5% oxygen. Obligate anaerobes can only grow in an anaerobic environment. Anaerobic meaning they do not require oxygen for life and for reproduction. It aero-tolerant anaerobes liwat is diri gyapira na requiring oxygen, but they grows better in the absence of oxygen. Pero pwede gyapira makag-survive in the atmosphere na may the oxygen. And lastly is the facultative anaerobes, meaning they are capable of surviving in either the absence or the presence of oxygen. So next characteristic is the nutritional requirements. So pareho si tao, bacteria kailangan gyap nira mga elements or mga nutrients para hira mag-grow nga makapag-survive. So like for example, those elements na carbon, hydrogen, oxygen, and etc. So they are required by some bacteria. May daliwat mga microbes na may dala nila specific na vitamin na kailangan. So some need organic substances that was secreted by other living or microorganisms during an era growth. So may daliwat mga organism nga fastidious na demandiran nutritional requirements. Another is the biochemical and metabolic activities. So, ang bacteria is na-identified through an enzyme nga ira ginsisecrete. So, this is useful also because through this, nadidetermine or na-identify kung what kind of organism is able to break down a certain substrate. Another is the pathogenicity. So, pathogenicity, it is the ability to cause diseases. So, many pathogens are able to cause diseases. For a reason nga, they possess capsules, villi, or they secrete toxins that damages cells and tissues. And lastly is the genetic composition. A genetic material is unique to another species. So with the help of the genetic composition, nadidetermine whether an usang organism or usang species is related to another. So now, i-discuss liwat natin ang mga unique bacteria. So what are those unique bacteria? So they are the Rickettsias, the Chlamydias, and the Mycoplasmas. So they are closely related to bacteria. Pero they do not possess all the attributes of a typical bacteria cells. That is why they are called unique. So Rickettsias and the Chlamydias are bacteria with a gram-negative type of cell wall. So they are obligate intracellular pathogens kay nakosiran diseases sa humans and other animals. So they must live within a host cell. So tanan na disease nga caused by Rickettsias are arthropode-borne, meaning they are transmitted by arthropode vectors. So, ato na mga arthropods are lice and mga fleas and ticks which transmit the ricketias from one host to another by their bites or waste products. Well, the chlamydias is referred to as the energy parasites. So they can produce adenosine triphosphate molecules, but they use ATP molecules produced by their host cells. So it chlamydias is transferred by the inhalation of aerosol or by direct contact. So it mycoplasmas is they do not have cell wall. So kay tungod na waray ra cell wall, they are resistant to treatment with penicillin and other antibiotic that works by inhibiting cell wall synthesis. So it mycoplasmas nakakos hiyahin, pneumonia ngan genitourinary infections. So let's proceed to the Archaea. So Archaea is a prokaryotic organism like the bacteria. So Archaea is more closely related to eukaryotic organisms rather than bacteria. Pero both archaeans and bacteria are prokaryotic as I have said earlier. So archaeans na differ bacteria in several ways. Kay it archaeans, they possess a different type of RNA. Thus, cell walls, they rin na containing peptidoglycan. Ngan, many of them live in extreme environments, and some produce methane.